All right, so it looks like we are broadcasting now. This is the Microbiology Journal Club, and this is our last book club discussion of the summer. We could still technically say it's summer, I think, because it's early September. So even though most of us, I think, are now either starting or well into our semesters, this is the last of our summer book club before we transition back into being a journal club instead. And so today, we're going to be talking about this book, which I think has been really rocketing around the Twitter sphere for sure. So I'm sure that people have seen this mentioned. This is Ed Yong's new book, very much new, hot off the presses. It came out, I think I got my hands on it in like early August. So it's I Contain Multitudes, and it's about partnerships between animals and microbes. So before we launch into talking about the book, I do want to say one thing really quick, which is we are transitioning to a new platform. So Google Hangouts on Air is dead. So everybody take a moment for Google Hangouts on Air. I'm so sorry. And then now we're on YouTube Live. So it looks like everything ported over well. The Google Hangouts format is still here. I guess they're just going to call it YouTube Hangouts now. I have no idea. But hopefully all of this is working well and everybody can see everything. So if we have any kind of tech going, we'll just blame it on the platform change. Okay, so what we'll do is really quickly introduce ourselves. My name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island, and I am currently teaching the non-majors microbiology course, and we are making our way kind of piecemeal through this book. So I told them I'd be interested in hearing what they think. And as we're talking about it, I'll probably talk a little bit about the perspective of using this in the classroom, which I'm very interested in what other people think about that as well. And next to me, down below, we've got Nicole. Nicole, do you want to introduce yourself?
I will, now that I'm unmuted because my screen is lagging again. Sorry. I'm Nicole Suktio, and I am a postdoctoral fellow at the University of Northern British Columbia in Prince George, British Columbia, where I have spent most of my time since 2013 learning more about characterizing soil, fungal, and bacterial diversity using DNA community profiling methods. And right now I'm trying to write up some results on early disturbance indicators from the fungal side when you shallowly excavate soil.
Very good, thank you. Oh, so you're in the process of writing, so hopefully we'll, I know that's a long process, but hopefully we'll see something that we can point to from you
Yes,
soon.
hopefully.
Very good. I think we had Kat and then we lost Kat. So this could be, we're going to blame it on the platform. That's what it's just, this is going to be blame YouTube Live Day. So instead, I think we still got Karen with us, although she looks a bit frozen in the little panel. So Karen, are you still there?
Can you hear me?
I can't, oh, there you are, perfect.
Okay.
Yep, I can hear you and see you just fine.
All right, I'm Karen. I'm just south of Nicole at Simon Fraser University. Oh, okay. And I study bacteria called Francisella that causes the disease tularemia. And I look at the proteins that it uses to cause disease. So in Ed's book, he calls them virulence factors, which is what are what I study. And so in his book, he talks particularly about the militaristic terms that are used to describe pathogens, and that's common vocabulary that I use in the way that I write or in the way that I describe things. So this was particularly eye-opening for me in thinking about, like, oh, okay, the vocabulary that I'm using can actually affect how people see these relationships, and so I found that to be very useful.
Yeah, that's a great point, and especially from the perspective of somebody who's actively studying a topic that gets covered in the book. So I'll be really interested to hear more of what you think about that, Karen. So that's awesome. And not to put her on the spot, but it looks like Kat just joined us again. Are you? Do you want to introduce yourself? It's Blame YouTube Live Day, so if something went wrong, you could just blame the new platform. So Kat, do you want to unmute yourself? Oh, maybe. Let's see.
Is that
So I don't, oh, there we go.
Hi. I will totally jump on that bandwagon and blame them. Yep, so here's my I contain multitudes, and I just have to show this. This is my signature. Thank you, Ed. I'm an assistant professor from the University of Alaska Anchorage, and I just got back from the ASM Beneficial Microbes where Ed spoke, and I'm really excited to talk about this this book. Thank you.
Okay, thanks, Kat. Did you just get back? When did you guys? I know that meeting ended recently.
I got back at 11 o'clock last night, so I'm waking up.
Oh wow! Well, thank you for joining us, even if you're jet lagged.
Yeah.
A
little bit.
And
I forgot to mention I studied the gut microbiota of stickleback, and I'm interested in host microbe interactions.
Excellent. And as Kat mentioned, if you've been following ASM Beneficial, so that was a very recent meeting where basically it was, a parade of people that were featured in this book. And it was really great. Thank you, Kat, and other folks for tweeting that, because it was wonderful to be able to follow along. I didn't get to attend, but with people live tweeting, it was really good to be able to see all of that. Okay, so without further ado, this is the lengthiest book that we read, and it is a very impressive work of synthesis about what we know and where we're going with looking at how microbes are living in relationships with animals, how microbes affect animal health and development. And it just is a pretty amazing synthesis of a lot of the current work, especially in the case of things where some of these topics, there's a lot of nuance, and there's a lot of what do we know, what don't we know. And I think he's done a really good job balancing amongst those kinds of the data that's available, the questions that are being asked, where the field is trying to go to kind of refine our ideas about microbes and their impact on animals, on us. Before we kind of get into details, I thought I would just ask, that was my general impression of the book. What was your general impression of the book for the other folks here? What did you guys think of it?
Okay, so I think the context is everything message was extremely clear and extremely played with without heavy-handedly saying it explicitly, which never actually happens in the entire book. But I mean, even starting with the idea that the connotational lens of looking at microbes initially has always been through pathogenicity effects, recognizing that the swing towards also just diversifying the polarities by saying there's good microbes and bad microbes, while recognizing that physiologies, the length of time for interaction, and some of the chemical ecologies and some of those like more microscopic or molecular interfaces that are dictating how a bacteria is engaging with a host, be it in an extended symbiotic relationship, be it in a developmental maturity context duration of the relationship, that it's very multifaceted, and there's some fabulous experiments that I as a researcher have envy for not being part of those narratives that play out that nicely or play out that convolutedly, but the idea that we really have to go case by case, and that's number one, sort of the beauty of the insight that microbiome type methods have brought us, as well as the blocks that we have in terms of policy and translational aspects to consider from these types of data collection. And that's what I have to say about that for now.
Yeah, I think those are really good points. I absolutely agree with you about the context thing. Kat, did you have some thoughts?
Yeah, one of the things I really liked about this book is that it's so accessible, that it's written really for the general public. It covers so much information and covers just, it's a really good history of everything that's been done, but it's not just a history of what was done, it was the stories behind the history. So where did this person come from, and why did they focus on this, and what makes this a really interesting story for people who aren't scientists to be able to get interested in it. I think that's what I really liked about this. A lot of the history I already knew from being in the field for a couple of years, but the stories behind the discoveries, or why one person got interested in this particular system, this particular squid, and this particular microbe, I thought those were really engaging and helped bring other people in. And in fact, I had people asking me, oh, can I, I see you're reading this book, what do you think? Can I read it? I'm like, well, actually, yeah, I think most people would be able to understand this book. And that's not something you see in every science book.
Yeah, that's true. I definitely agree with you. I have a thought about that too, but I don't want to get too far ahead of myself before Karen, if Karen has anything she wants to say kind of generally as an introduction.
There we go. Okay. Yeah, so I guess think two just things to build on what Nicole and Kat has said already. Just the fact that Ed uses this language that is able to balance between like good and bad and the complexity of relationships. And one of the things I enjoyed that he talked about the mathematics of of how we normally think of healthcare. So it's like it's an add or a subtract thing. Like, okay, you have this disease, so then you do X to treat it, or you have this problem, okay, let's just get rid of that. And so the simplicity of how we're treating healthcare is not reflective of the complexity that's involved in this entire approach that we need to use to look at it. And I think that Ed really clearly illustrates that in the book, that it can't just be, okay, it's good or it's bad. It's very much like relationships that we have regularly as a whole, where you have a symbiotic relationship with your family, but then at the same time, like those same relationships can also be detrimental or difficult. So it's never that, oh, okay, it's always purely good in one context or purely bad in another context, but it is a symbiosis of sorts. And that's what I really liked about it, that he was able to highlight that complexity that is absent in a way that very much of the time when we teach science, we don't. It's hard for us to teach complexity because I think at the same time it's difficult for students to grasp complexity. And I think that in that sense the book is a really good gateway for students to start thinking about those things.
Yeah, I absolutely agree with that. I think that's because I'm teaching this book, as I mentioned. I'm teaching this book right now in my course for who are not science majors. And I'm really trying to get them to think about one of the things we keep coming back to, which I think Ed gets across really well, is we have had a tendency up to this point to think of things as kind of a single microbe problem, which he mentions explicitly when he's talking in a certain section about disease. So the idea that, and this, of course, it's grounded in truth, right? Because if you die of Ebola virus disease, that's because you had Ebola virus. So when you've got Koch's postulates that have been tested, and you can see these causal relationships, single microbe in a disease that manifests, it makes sense that that's how you're going to focus your efforts and your thinking, is on a single microbe that comes in and causes a problem. But what I really like is he's trying, with all of the examples and the discussions here, to get the readers to think about your microbial communities and your relationship with microbes as an ecological problem and not a single microbe problem. And this gets mentioned in the context of disease when you're thinking about chronic ailments and autoimmune disorders, that maybe we need to be thinking about communities that are out of balance, or that there's been some disruption, rather than trying to identify individual villains that are causing this problem. Maybe it's just a community on a larger scale. But just today in class, we talked about prebiotics and probiotics. And the same thing applies in that discussion, where right now the probiotic industry is kind of focused on eat this yogurt and it has Lactobacillus casei in it, and that's going to be great. But truthfully, as he mentions in the book, what we probably need is a more thoughtful way of thinking about microbes as communities and networks that work together. And that if you're going to take an effective probiotic approach, you have to know a lot more about the network of microbes that are going to be acting in order to get the kind of result that you want. And that's been great for the students because that's kind of a broad idea that gets at the complexity, but it's something you can kind of keep trying to bring them back to, is how are we moving from a single microbe viewpoint to an ecological viewpoint. To try to get them to think on that scale, I think has been really good. So I don't want to, yep, Kat.
I wanted to piggyback on that, and I think in addition to thinking of microbes as a community, he also did a great job of laying out the argument behind systems biology, which is that we don't just think of a single microbe interacting with one aspect of the host, but it's a complex relationship where the community interacts with a number of genes and a number of parts of the immune system or a number of metabolic processes, and that this is when we look at these relationships, we have to consider a much bigger story than just the way that we've been thinking of microbes and hosts in the past, which is this one microbe produces this one toxin, which elicits this one part of the immune system, and this is how we fight it. It's more like this is a complex microbial community that is producing a lot of different metabolites and interacting, increasing and suppressing the growth of several other organisms and interacting many different or activating many different parts of the host immune response and metabolism and all these other things. And it's a much more complex relationship than we initially started studying it as. And I think he does a really nice job not simplifying it, but conveying that message while not making it so complex that we get overwhelmed by all of these interactions. And as a systems biologist, I really appreciate that.
Yeah, I agree with that. And I think this in similarity to Carl Zimmer's book, I think there was a good attention to being careful about jargon. So he was good about giving the full Latin name for these microbes, which tend to be long and complicated, but then they get shortened, and it's a very conscious effort, which I imagine took a real effort in order to get it organized in a way where you're not throwing too many acronyms or too many unfamiliar terms at people, because you've really got to try to keep them straight. That's one of the things I've noticed with students is you can, like if I've talked to them before about Vibrio fischeri and squid, and you kind of have to continually remind them Vibrio fischeri is a bacterium. It's a bacterium. It's a bacterium, like, because they just forget, and they're like, Vibrio fischeri is the squid? Is that the, no, it's a bacterium. Like, so I think he did a pretty good job trying to keep everybody tracked through with truthfully, a part of biodiversity, the microbial world, that a lot of people aren't really familiar with and don't really think about. So already he's kind of tackling that, and I think he does a pretty good job trying to keep everybody on track with what he's talking about. Nicole.
So I guess I'm going to pick up on the themes of accessibility to readers, and especially since you are actually doing the hard work of teaching well, teaching in general and to non-majors with this book. I think it's a fabulous idea, and I think I want to start thinking about what you've thought about concept mapping with the book, because it is very well written, but it's lacking the compartmentalization that Carl was able to get in his book by only focusing on one domain of life and having a much more schematic layout. So one thing I will say that I thought was really handy if I was going to use this to teach with non-majors was actually chapter 2, because even for myself, going back to my own narrative of how I got back into sort of 16S and ITS2 profiling, at the time that like a lot of the earlier studies were coming out with Ed DeLong and Norm Pace, I was a protein biochemist, and I have to say that chapter 2 is fabulous for like going all the way back to Leeuwenhoek's, you know, observations of the actual organism to describing how we started using phylogenetic anchor sequences without using that jargon of phylogenetic anchor, and explaining why DNA became such a powerful tool for looking at, the microbial world around us. So I guess I'm kind of wondering whether you, Laura, have anything to say about what you've been doing in terms of making a roadmap out of I Contain Multitudes. But obviously there's other things to talk about in the book as well, so not too much, I guess.
Yeah, so this, for some reason, I just had a flashback to, like, political forums where they ask them, like, a long, complicated question, and then they'll be like, briefly. And I'll be like, oh, okay. Yes, I agree with you. I think I'm going to be completely and totally honest. This length of a book, the non-majors are not going to make it through this in the way that I teach the course, because we do the microbiome in the first third. Because it's a microbiology for non-majors course. And the way I currently teach it, it splits up between the microbiome to start with, so microbial partners. And then we have a section called microbial threats, which are about disease, the stuff they're more familiar with. And then our final module is microbial technology, which is things like food and drink, bioremediation, biofuels, things like that. I really, really want to teach an entire course on the microbiome for our majors. And I think this book in particular we could definitely make it through the whole book if you had a course devoted to this, which I think would be great. Considering what I have to work with, what I've been doing is tracking along with my existing course material. And then I found that much of it, which is kind of reassuring to me, I have to admit, I was like, oh, good. It parallels really well with a lot of what he's talking about in the book. So I'm able to kind of lift sections and have them kind of go through the book in sections. So I will say that the book is if you approach it from that perspective, if you're not going to read it cover to cover in a class, he still does a really good job splitting into sections. Here's what we're talking about. Here are my illustrations. Here's the concept again. And then you can move on. So I found, fortunately, that the book is structured really well, that you can actually lift individual sections. They struggle a little bit with why we're outside of humans. So I did notice that. They're kind of like, and we're talking about coral reefs, why? And you kind of have to remind them, model systems, what can we learn from squid, why is that important? But for non-science majors, that really throws them. They're like, now we're suddenly not talking about humans. I thought this was a microbes and man course. Why is it not about me? And so you kind of have to bring them back a little bit. And so I think this is a good test run for me. And I think we'll see how it goes. And I'm really curious what the students think too. They seem to be picking up on the things I want them to pick up on, because I'm having like questions and forums for them to have answers. But we'll see. That was my not so brief answer. Does anybody else have any thoughts, having read this from a potential like teaching perspective?
I just wanted to say that what I liked about this is that it's not just accessible to people who don't study the field, but people who would like to know more about the field, who are already in the field. Because one of the things that I noticed as I was reading along was the notes in the back. I'm kind of a nerd. I read the notes in the back of books as well. And the text is really accessible to the general public. And then if you want to know more about the history of the word microbiota, or this particular scientist, and a little more in depth about their studies, you can go to the notes, and it's actually a really great collection of things that maybe the general public isn't going to want to read, and they might just read the text, but those of us who are really into this information can get a lot from the notes as well. So I could see teaching this as for I'm not sure about non-majors, but I could definitely, well, maybe teaching this, using this book to teach lowerclassmen and upperclassmen, and having upperclassmen expand on some of the ideas that are brought from the notes in the very back.
Yeah, I agree with you. I thought the notes were fantastic. And I do want to say, just as a platform note, I can't tell if I'm lagging or not, so I'm sometimes pausing quite a bit because I don't want to cut anybody off if they have something to say. So, but I agree with you. I think the notes were fantastic. I'm kind of curious, but just from a behind-the-scenes writing perspective, if that was the structure to start with, or if this was a structure that came about through input from editors and other readers and so forth. Because I've read a lot of Ed Yong's work when he's been writing pieces right now for The Atlantic, but then in the past when he was at different places. And I think I didn't sense that his writing style changed that much from those to this book. But I do kind of wonder, how did you decide to put something in a note instead of try to work it in? Was there a draft and all this stuff was in, and they're like, no, we better move this? But that's more, like I said, a behind the scenes thing more than anything else. But yeah, I agree. I thought the notes were fantastic as well. So one of the things I wanted to ask about with this, and another kind of theme that we've touched on a little bit is this is very complex, but then also a lot of what is getting covered in the book is very current. So, you know, there's somebody I know who calls this like the bleeding edge. So it's not like the cutting edge, you're actually on the bleeding edge of things. And one of the topics covered in here, for instance, is the hologenome, which if you're in the field, you may be aware that this is a concept that certain people are big proponents of and certain people are not. And there's an ongoing discussion in the field about the utility of the hologenome idea, what it can tell us, where its limitations are, is it necessary, and that this is an active source of debate. And that's one example of how he dealt with uncertainty. So that's kind of something that gets touched on in sections throughout the book, is when you start to push up against something where the field is not agreed, there's not consensus. And so there's uncertainty. So for instance, bacteroidetes and firmicutes. I'm going to teach this tomorrow in my non-majors class when we talk about the microbiome and obesity. There's a mouse experiment where they show the ratio flips, but if you look in people or if you look in other mice, it doesn't always do that. And so he spends some time kind of talking about uncertainty in terms of what we know and what we don't know. And I just wondered if you guys had any thoughts about how that was handled. Do you think that was conveyed well? And there you are.
So I think I have a few thoughts about that. And the first was that I have to get props for being so articulate and clear about these ambiguities, but finding a way to write about them that does not undermine the credibility of the work done. Because one of the biggest challenges, and actually this is going to deviate a little bit from the question, but it's going to come back really quickly, I promise, is that he's really helping with the image problem of scientists. They're written up as real humans with other interests in this book. They're written up as people who are willing to say they're uncertain about the significance of their findings or what their mental map of what the overall causal or underpinning mechanism for what they're seeing is. And that's a very important thing to have articulated in literature that's coming out, that's designed for the broader public to understand why science as a profession doesn't translate into something that is marketable in 5 years. It doesn't translate into a clear direction for a policy or a conservation approach or a therapeutic approach. And with respect to comprehension, it will leave an audience potentially at as much of a loss as the scientists are for some things, and that's okay. But the other brilliant thing he does about that, and I particularly like it in the chapter called In Sickness and in Health, is that there are a lot of, first of all, he uses a lot of really excellent analogies that can be even used, drawn on a board, used to set up a lecture, but he's pulling in other concepts from other empirical studies of disease, other schemas used in ecosystem ecology that you can map these microbiome studies onto. So for example, in Sickness and Health, I think around page 138, he actually uses a description of the adaptive cycle, which is a common paradigm, well, or scheme of thinking about ecosystem progression and succession, I just put up a link that actually shows the diagram in relation to reef health. But then he also uses it to map onto the idea of gut dysbiosis, and I love it. Towards the end of that chapter, he also discusses the limitations of microbiome studies with respect to small n's, with respect to, again, plurality of outcomes, with respect to length of study, with respect to not using humans as the model. But then he talks about the idea of, are we going to use multiple sampling points? Are we going to study things that would be an age-matched cohort, which I think is awesome, because now he's actually setting up, if you were using the book for teaching, and also if you were reading particularly from a public health epidemiology model, how you would be mapping a microbiome experiment onto pre-existing research designs that have been considered gold standards for assessing risk reduction, and refining hypotheses around why certain factors, in this case microbiota, lifestyle interaction with microbiota, may translate to a disease state or a non-disease state, or a constellation of complex symptoms that may or may not predict a disease state. So that's what I have to say about his handling of complexity.
Yeah, that's great. I completely agree with you. I thought that's very well said, and that's a number of really good points to make about that. I'm going to ping to Karen because she's kind of frozen on my little panel, so I think she's still there, but I just want to see if she has any thoughts about the uncertainty, handling uncertainty.
I didn't have any, like in terms of handling uncertainty, I guess my question is because I'm not in the Halo biogenome field, so I don't have a perception on what the proportion or distribution of people that agree or disagree looks like. So the way that Ed has laid it out in his book, he's laid it out, okay, there are three different perspectives, which are, there are people who are strong proponents, there are people who are kind of in the middle ground, and then there are people who are highly against it. So, well, I did have a question, which is, in terms of you guys that are in the field, is there a strong perspective that dominates the field in terms of the spectrum of the three, or is everybody really just evenly split over the three? I don't know if you have any thoughts on that.
I'd be interested in what Kat thinks about this too. I think she's back. But my impression, and this is honestly just from watching discussions happen on different social media sites and seeing there's been a bit of an exchange in the literature as well. My impression is that everybody right now is kind of, most people right now are kind of somewhere in the middle where they're looking for more information. And that you've got people like Seth Bordenstein, who is in the proponent camp, and then you've got some folks who are in the opponent camp, or I don't even know if I would call them strong opponents. They're just more saying, like, is this a new idea that we need to articulate, or is it saying something we've already said? I feel like the major opinion right now is we need to define it clearly what we're talking about, and then we need to start collecting some data to figure out what it is that to refine what we're talking about. And that seems to be where a lot of people are sitting, is they've got this idea and now they're kind of pushing up against it and going, what about this? What about that? Dave Baltris, who joins us sometimes but has had kind of a crazy summer, is one of those people who will basically take an idea and just be like, push, push, push. What do you how do we stretch this, and where does it go? And what can we make it do, and what doesn't it do? I feel like that's where we are. Kat, do you have thoughts on that?
I think that's a really good description of where the field is at right now, where people are debating whether or not we actually need another word or another mechanism for studying these. We already have people who are studying microbes and their interactions with the host, and whether that host is a plant or a bug or a person. And so do we really need this definition of this? Some people are kind of think of it as like maybe it's too narrow of a definition, or maybe it's too broad of a definition. So I think a lot of the discussion has just been around what is the exact definition of holobiome and how is that different from the other definitions that are out there. You know, why not just call it systems biology, or why not, we're already doing these studies. Do we really need to have this be another focus area? And I really like it because I like these philosophical decisions, or these philosophical discussions about where a field is, and whether studies need to be done and what studies need to be done. And then I really, really appreciate these discussions around the vocabulary, because the vocabulary really does change the way that we think of something. I go back to the definition of a pathogen and a commensal by Arturo Casadevall and Elise Porofsky, where they talk about this relationship that we have with these microbes, where we used to call it a pathogen, and then we used to have this clear definition between pathogen and commensal, and then we made it a little more fluid, and we started studying these relationships with the microbes and hosts in a different way, because we realized that a microbe could be both a pathogen and a commensal under different environments, and that's changed the way that we study it. And I think that the idea of the holobiome is the same thing. You know, we're trying to define this. Maybe it's something that we already kind of have a definition for, and we just need to. I'm not sure if we need the new definition, or if we just need to acknowledge that what we're already working on encompasses all this. And yeah, it's an interesting philosophical question. I'm enjoying watching the conversation. In the meantime, I'm just plowing ahead with looking at host microbe interactions and how the genetic background contributes to these interactions, and whether or not I'm talking about the holobiome or just referring to it as interactions. It's still the stuff that I'm interested in. So I think that's where the field kind of is that we're going on with these studies. The definition might change. We might use a new word. We're not sure if we need this new term or not, but it's a fun discussion to watch.
Was any of that discussed at the meeting that you were just at? Did it come, because that seems like an appropriate place for it to come up if it's going to come up. Did the term, are people using the term when they talk about their work? Or was there any discussion between attendees at the meeting?
I do remember the term coming up once or twice, but it wasn't the main focus of anybody's talk. One of the things I would like to see for this conference and other conferences is a speaker who just focuses on definitions and philosophy of the work that we're doing. Everybody has a 15 or 20 or 30-minute block in which to describe all the work that they're doing in their lab and everything that's going, all the background you need for it and whatnot. And so we don't tend to get into definitions very much. The definition between commensal and pathogen was brought up a couple of times by people who are studying microbes that cause disease and their interactions with the microbiome. Holobiome came up once in somebody's definition, in somebody's introduction, but it wasn't really heavily discussed. There might have been smaller discussions in between people over coffee and whatnot, but yeah, we didn't have that much of a discussion. I would love for that to be a section at one of these meetings.
Yeah, I think that would be good too. I think that could be very valuable for pushing forward some of these conversations rather than having it happen in our 140 character limit tweets or, through the literature which tends to be delayed. That is a really good idea. So I didn't want to cut anybody off. If anybody else has any other thoughts about uncertainty? Good. Okay. All right. Feel free to jump in if for some reason my YouTube Live platform is cutting you off. One of the things that I wanted to ask about with the book too, and this kind of sort of deals with uncertainty, but from a different perspective, I guess. There is, we're talking about antibiotic treatments and the impact on your microbiome, and what should we do about that, or how should we think about that? That's something that we talked about today in my class. We're going to read the section of the book on antibiotics and where Martin Blazer comes into it. We're going to read that for a class discussion that we're going to have next week, Tuesday. So I'm going to be really interested in what the students think about antibiotics and the deployment of antibiotics on the basis of having read all of this stuff. But that's one example. But basically what he's got here is a very kind of, I'd say, delicate presentation of, you've got Martin Blaser, who has really come to the forefront as somebody who's going to, he calls him, I think, like a microbial Cassandra, where he's just going to put out the idea that, you know, we're eliminating certain microbes from our microbiome, and this is going to have disastrous effects on us. We're going to see all of these long-term health problems and the rise of these epidemic Western diseases because the changes in our microbiome and what we're doing to them. And then he kind of brings in Jonathan Eisen and the Overselling the Microbiome Award. So you get these, it is interesting. I really wish from one perspective, I could read this book without the knowledge that I have, because I'd be curious as to what a completely naive person makes of all of this. I'm coming at it having seen these exchanges happen on Twitter and seeing Jonathan Eisen kind of put out the Overselling the Microbiome Award. So I've got a little bit of baggage, I guess. We read Missing Microbe, Karen and I did. And so I was kind of curious, like, how does that, when you're talking about something like this, where you could, and this kind of gets back to some of Nicole's points, where you could be forming policy on making decisions about, so the FDA just last week, I think it is, said they're going to start banning triclosan from any products, any commercial products. So if you buy hand soap with triclosan now, I think the FDA is implementing a ban. I don't know the particulars of how that's going to roll out, but they've kind of finally made a decision to say, like, you know, no, this is ridiculous. All this antibacterial stuff, we don't need it. How did that part read to you guys? Because I think that's a big place where policy and healthcare and people who, like Martin Blaser, who are almost evangelical about their interpretation of their data. How did that come across to you?
So I think for me, this is it's part of why conservation biology, and attempting to have a conversation about it, especially since I'm not a conservation biologist, but Martin Blaser is talking about conservation biology. It is a very value-driven, context-dependent, connotation-associated discourse in general, as well as how the grounding of what a conservation effort is. I mean, if we're talking about Western diets, Western lifestyles, and the fact that most of our data has come out of probably Western pseudo-epidemiological studies of the microbiome, where are we going to ground the idea of what dietary modifications have to be made? Is it really about trying to approach or shadow diets from hunter-gatherer tribes, which I don't think is really going to be the answer in terms of what the current landscape of food availability and norms of purchasing are in North America anyhow. So the idea about extinction events is interesting, because this is not the type of extinction event that gets press, you know, but it's also a very key one to think about as we go forward in time. What are the health effects of data diversity that has never been part of mapping onto the longitudinal studies that have been done in the States, which are like largely, you know, looking at heart health outcomes, very physical growth data of children for something like Framingham, where you've got like a bunch of people who are followed over time for their weight, for certain, you know, as a look at, as a glimpse at health and nutritional status. So we're mapping this on late, and I think it's actually really too early to say that we know translationally what we need to be doing about alpha and beta diversity of gut and non-gut microbiota of people. And I mean, that's sort of the hand-wavy answer that I think the public doesn't like. But I mean, we have to remember that we've really only been characterizing these, and they might just need to be mapped onto some of our existing study designs, where if you have like a twin-based study or an age-matched case control, that we can at least calculate probabilities of a disease outcome based on what we're maintaining and not maintaining. So I think it's actually really cool that Ed's kind of got the eyes in versus blazer idea, because it's also making the idea of how complex thinking about what to conserve and how socially, politically, and value informed that decision is made, whether it's pandas or whether it's stuff that's in our own colon, which is what I find kind of fascinating about it.
Yeah, I think those are some really good points. Karen or Kat, did you have thoughts on that?
I was going to talk about like how my perspective evolved because I started with missing microbes, and that was the first time that I'd come in contact with the philosophy and the ideas that, you know, eliminating certain species in our microbiome would be catastrophic. And so from that perspective, like I guess even personally, like I would develop this bias based on this initial perspective that I had discovered. So then when I encountered the social media different conversations of when Jonathan Eisen tweeted, like that, when Jonathan Eisen tweeted the, what was it, the award? I can't remember the name, but
yeah, it's overselling the microbiome.
The overselling microbiome award. Like when you tweeted that, I was like, oh, so that's not because I had only read Missing Microbes. It was like, oh, there's something else out there. And so, and the initial reaction for me was like, oh, does that mean that Martin Blazer was wrong? Or like, I had to go through this entire process of becoming okay with these multiple perspectives and realizing the complexity and the diversity, and this journey wasn't just isolated to these particular concepts. It's also part of my evolving as a grad student, like becoming an understanding that there's biology is extremely complex, and most of this complexity is okay. I guess the problem comes like because we understand that complexity is okay, but the problem comes is if complexity is okay, how do we take action based on the fact that complexity is okay? And I think that's where policy and the public are more apprehensive, because complexity is okay does not lead to a definite action. Like so if this is okay, then that means what do we do about it? And they're just kind of at a loss. It's like, so if my kid has this, does that mean that I take, does that mean I give up probiotics at this age? Does that mean that I do nothing? You know, they're at a loss because they want something to do. They feel that doing something will help them improve their lives. And that's what they're looking for. They're looking for that end thing to do. And so even personally, I haven't found the balance. Like the one balance is, okay, don't do anything until we figure out more. I mean, that's certainly that's one plausible action, or we can cautiously do like small things until we know more, or experiment. And and so as that journey is happening through me, I can kind of understand how how everyone else's perspectives are confusing, and they also have to walk through this journey. And maybe some people never get there to the end where it'd be like, okay, it's complex, and you know, you just have to try it, and if it works, great. If it doesn't work, then we try something else. So I think that as a grad student, I walked through that journey, and as the public has to walk through that journey too. So
I think that's very true. I think that's, yeah, kind of the crux of the problem. Kat, did you have thoughts on that?
I just wanted to say I really like the way she said that she is a grad student. Karen said that as a grad student, she's had to walk through this journey, and as the public will have to walk through this journey too. And I think that this book is a good step in that direction, and that another discussion that's often had on Twitter is what our role as scientists is in making sure that the public understands the complexity. You know, it used to be that we could just treat an infection with penicillin, and then we were able to show that we couldn't treat all infections with penicillin, and then we had to educate the public about multi-drug resistance and testing testing microbes for antibiotic resistance and treating specific microbes with specific treatments. And I have a lot of faith that the public is going to be able to wrap their heads around personalized medicine and start thinking about, okay, not every treatment, not every disease is going to be able to be treated in the same way. And I think this book is a really good start into pushing for that. The other thing I wanted to comment on was this idea that, you know, science is kind of slow, but there are definitely studies out there that are showing that there's, yeah, we don't want to overhype the role of the microbiome in health, but there are studies that show that, you know, there are certain things that are beneficial, and we're having to fight this balance where we're having to strike this balance where we want to get the science out there really fast, but then we don't want to overhype it. And we also want to make sure that all the proper controls are done. And I think that this is going to be the next step of microbiome studies. It's just making sure that everything that we're doing is presented in such a way so that we're presenting the complexity without overwhelming the general public that we're trying to help with these studies.
Yeah, I think that's very well said as well. So everybody had really good points. That's all of that. I agree with. Yeah, and I think I agree that I've seen the push towards having more defined standards, so that groups who are doing microbiome studies, it's a bit more controlled across labs, and it'll be interesting to see how that evolves. You know, I don't know if it's going to get as extreme as this is the genomic DNA prep kit that you should use. Like, companies would love that because then they get a monopoly on who sells like the genomic DNA prep kits. But trying to get these a little bit, make sure everything is rigorous, and so that you can make cross comparisons and have something meaningful to say. I think that's going to be important. So we're doing some microbiome work in my lab, so I'm definitely thinking about that, making sure that we're doing all the proper controls and that we're being careful and rigorous with our data. And so that's been interesting to see that from the actual wet lab computational workflow side of things. Okay, so we're coming up on 4 o'clock. Oh, Kat, please. Yeah, I thought I saw you on mute. Yeah.
Yeah, sorry. I just wanted to say that that ties in really well with where we started the conversation with, early on in the conversation, we were talking about how cutting edge this book is, because some of the things that the conference just yesterday were things like looking at the how not just one mouse species or population responds to this treatment, but how does it do, how do different mice from different vendors who may have different microbiomes or who come from different facilities, even within the same campus, like people are starting to incorporate these in their studies. And I think that was not something that we considered even, you know, 2 or 3 years ago. Where did your mice come from? Well, they're all Black 6 mice, but this one comes from Jackson Laboratories and this one comes from a different company. And how does that affect their microbiome? Well, now, just 2 years later, we're already starting to incorporate stuff. And that's a really fun thing about this book, is seeing there are things that he brings up in the book that you're like, oh yeah, now that's almost standard. And this was, it was something that we didn't really think about even 2 years ago. And now we're thinking of it and incorporating it in our studies. And yeah. Another really big benefit of this book is just how cutting edge it is.
Yeah, that's true. I can't imagine trying to write this. I mean, I'm overwhelmed trying to keep up with the literature, and I'm not trying to write a book on a field where there's amazing studies published every time you turn around. And I seem to remember at some point on Twitter, he just made this like desperate appeal that was like, people please stop publishing things because I have to get this draft done of my book. So yeah, it was a monumental task, and it really catches this field at a really amazing time. And it's nice to have a text to be able to point people to if you have people who are curious about the microbiome because they saw it in the news or something, and you could say, well, listen, if you want to know more, then you can look at this because this is pretty current. You know, it's handled carefully and it's handled thoughtfully, and it's a scientific synthesis of a really great form. I'm planning to give this, they won't watch this, so I'm not going to give anything. Maybe they do. If they do, hi parents. They're going to get this for Christmas, so I've just spoiled it, but they probably expected that anyway. So we're coming up on 4 o'clock Eastern time, and I'm always reluctant to keep people longer than an hour. Does anybody have any closing thoughts about the book before we end our final MicroJC summer book club? I'm going to cut anybody off. Everybody's kind of paused. Okay, so if nobody has any closing thoughts, thanks for being here, because I really enjoyed reading this, and I've really enjoyed discussing it with you, because as we could have gone on longer because there's a lot of interesting ideas in here. So it was really valuable to be able to chat with you guys about this. And thanks for being here for the book club, because we've read 3 really awesome books, all 3 of which are going to make it into my class in some incarnation or the other. So that's been really beneficial for me, and I really appreciate that. Karen, did you have a thought? I see you're not got your microphone on.
I was just going to say, this is the first time that I think we've had 4 females in the book club, more than just Laura and I. And I thought that was worth pointing out. I think that's very cool.
I didn't realize that. Okay, well, there you go. That's very good. That's a good thing to end with, I guess. Anybody other thoughts, Nicole or Kat?
I was just going to say, if there are any entomologists and behavioral ecologists watching this, the book is actually dope for those 2 camps. Like, there's some really cool stuff in there.
That is true. Lots of love for insect endosymbionts, which I did my postdoc on, and Blackmania shows up a couple times, and I was like, oh, Blackmania. So, Kat, did you have thoughts?
Just piggybacking on that gender equality in the video, one thing that I was really impressed with this book is the gender equality and the diversity, not just of people, but the diversity of the models that he covers as well. And I think as a scientist, female minority scientist who studies a model organism, I read this book and I just kept wanting to cheer, like, yay, over and over again as I was reading it on the bus, on the subway, on the wherever. And worrying what people would think if they saw somebody sitting next to them in the airplane going, oh my God, that's amazing. I can't believe you just brought that up. So thank you, Ed Young. You did an amazing job with this book, and it's going to be a huge benefit for the field and for the general public as well.
Well, I don't think I could have said it better than that as a closing statement. So thank you, Kat, for that. That's a really good wrap up. And so thanks to Karen, Kat, and Nicole for being here. And we are going to be switching now to our journal club for the fall semester. So our next meeting will be in October. I don't have a paper picked out. Somebody might have suggested one in May, and I don't remember. So if somebody suggested a paper for us to do and we haven't done it, please suggest it again to me, because I don't remember, and my brain is already scattered, which is not a good sign. So we're looking for papers for our October journal club, which we'll be doing. We'll announce our date once we've got a paper, and we'll kind of try to pick a time that works for everybody. So thank you very much to everybody for being here, and thank you for reading the book. And we'll sign off now, and off.